Hello everyone, Mr. Linder here. Let's talk about cell-to-cell communication in the nervous system. So the nervous system relies on synapses, and synapses are connections between two cells. And it turns out that the nervous system will utilize both electrical and chemical synapses in order to communicate from one cell to another. A synapse really has two main parts to it. The synapse is going to have the presynaptic cell, and it's also going to have the postsynaptic cell. And when you look at information flow at a synapse, you're going to have information flowing from the presynaptic cell to the postsynaptic cell. Now, what's interesting is that the postsynaptic cell is not always another neuron. Oftentimes, we think about communication in the nervous system as neuron to neuron communication. So you would assume that the presynaptic is a neuron and the postsynaptic is a neuron. But the postsynaptic cell could actually be cardiac muscle, it could be smooth muscle, skeletal muscle, glandular tissue. So the nervous system is going to communicate from one neuron to another, but that postsynaptic cell can be other things as well. So whether it's muscle or glands, etc., we're going to have communication from a presynaptic cell to a postsynaptic cell. Another thing that's interesting, oftentimes when you look at diagrams, it seems like synapses are only between two cells. But it turns out that a system of cells within the body where you have synapses making connections could be a moderate number of synapses, something on the order of 10,000 connections, whereas some cells are going to have hundreds of thousands of synapses. So it's not just this typical one cell communicating with another cell. You can have tens to hundreds of thousands of synapses with some cells within the body. But oftentimes diagrams are only going to show you a limited number of connections because the diagram would get confusing. The first type of synapse to consider would be the electrical synapse, and that's what you're looking at here. The electrical synapse really is just a series of gap junctions from the presynaptic cell to the postsynaptic cell. So what we're looking at here are actually proteins that create what's called a gap junction. And these gap junctions are made of what are called connexin proteins. And so hence connecting the presynaptic cell to the postsynaptic cell. And so information is going to travel typically from the presynaptic to the postsynaptic. However, you could have some information flow go back the other way just because these are protein channels connecting the two membranes together. But typically when we talk about a synapse, we want to talk about information flowing from pre to postsynaptic. In the nervous system, typically you're going to see these gap junction connections, these electrical synapses in the central nervous system. And the nice thing about these types of electrical synapses is they can be really fast. You know, information can travel from the cytoplasm of one cell to the cytoplasm of the other cell very quickly. So whether it's ions flowing from one cell to another, that information can travel very quickly from cell to cell. You're also, though, going to see gap junctions in other places in the body. For example, smooth muscle cells have gap junctions. Cardiac muscle cells have gap junctions between them. You'll even see them even in beta cell. In the pancreas, they'll have gap junctions from cell to cell. So you will find gap junctions in other places where you want to have fast communication taking place. So the nervous system utilizes electrical synapses or gap junctions as a way to get information from one cell to another. And again, the primary advantage here is the speed at which you can send information and also the synchronization that can take place as you interconnect groups of cells together. The more interesting synapse, or in essence, the more complicated synapse, though, to talk about is the chemical synapse. And so oftentimes you'll see the chemical synapse depicted as an axon terminal connecting maybe to a dendrite on a postsynaptic cell. So for example, this is an electron micrograph showing you the synaptic vesicles in an axon terminal. So what we're looking at here is an axon terminal, and these are synaptic vesicles. So these are vesicles that we see in the axon terminal, and these vesicles are going to contain neurotransmitter. And so when you think about a chemical synapse, you want to think about the neurotransmitters that are released. So the vesicles are going to release neurotransmitter, and that neurotransmitter, that chemical, can communicate with the postsynaptic cell. So this, of course, the axon terminal would be the presynaptic side, and then the postsynaptic side would be the dendrite or cell body or other type of cell, muscle cell, glandular tissue, that is responding to the chemical that is released. So the vast majority of synapses that you see in the nervous system actually are chemical synapses. And then these, of course, these chemical synapses utilize various kinds of neurotransmitters to communicate from cell to cell. And that's what's going to give the nervous system some unique functionality, depending upon what type of neurotransmitter is being used and what receptor it's binding to, is going to give you varying kinds of functions within the nervous system. And so we can talk later on about norepinephrine or acetylcholine, GABA, glycine, dopamine, etc. These are all different kinds of neurotransmitters, and they all can be involved in these chemical synapses. So whereas an electrical synapse is using gap junctions, a chemical synapse is utilizing the neurotransmitter chemicals to communicate from the presynaptic cell to the postsynaptic cell. In order for this to work, the postsynaptic cell is going to have a receptor. So in order for the postsynaptic cell to respond, there has to be a receptor for that particular neurotransmitter. So if you're releasing norepinephrine and there's no receptors for norepinephrine on the postsynaptic cell, there wouldn't be a response. If you're releasing dopamine and there's no dopamine receptors, there wouldn't be a response. So depending upon location within the central and peripheral nervous system and what types of receptors are responding to particular chemicals, that's going to dictate what kind of response you actually get. So how are these neurotransmitters getting into the axon terminal in the first place? So neurotransmitters have to be produced, and then of course they're stored in vesicles and then released. So neurotransmitters are actually made in the cell bodies. They're made by the Nissl bodies, the rough endoplasmic reticulum. And we are essentially doing translation to produce proteins oftentimes, and then they're stored within vesicles and transported along the axon down into the axon terminal. And so oftentimes what you're going to find is you're going to find vesicles within the axon terminal containing the neurotransmitters that were produced. So this is a vesicle, and within that vesicle, you're going to have your neurotransmitters. So your neurotransmitters are inside that vesicle. So this particular neuron could be producing glycine or GABA or dopamine type neurotransmitters, putting them into vesicles and then moving them down into the axon terminal. So again, remember, neurotransmitters are produced in the cell body of the neuron, and then they're transported down to the axon terminal. Your polypeptide-based neurotransmitters have to be made by your essentially ribosome. So oftentimes you get obviously your rough endoplasmic reticulum involved, or just your free-floating ribosomes within the cytoplasm. And then you have to have axonal transport. And so there's this, what we call anterograde transport we're going to move vesicles. I'm going to draw in some vesicles here up in the axon. We're going to move these vesicles through what's called anterograde movement so that we have vesicles down in the axon terminal with our neurotransmitters stored inside, waiting to be released. But that's where the chemical synapse and the physiology comes into play. How do you trigger the vesicles with the neurotransmitter to exocytose the neurotransmitter at the synapse? How do you get the neurotransmitter released into the synaptic cleft? Because you can't activate the receptors, so we need receptors on the postsynaptic side. You can't activate the receptors unless you release the chemicals from the axon terminal. So there needs to be a mechanism for exocytosis of the neurotransmitter so that it can diffuse across the synaptic cleft and therefore bind to receptors on the postsynaptic side. And so I want to walk you through how we actually get release of neurotransmitter at a chemical synapse. Now, sometimes smaller amino acids, smaller neurotransmitters, your amino acid based ones, could be synthesized within the axon terminal. But for the most part, we typically produce neurotransmitters in the cell body and we move them by anterograde axonal transport down to the axon terminals. But there are instances where you can make some of your smaller neurotransmitters within the axon terminal. So how does this mechanism work? And there are diagrams that are complicated and some are simplified. Oftentimes, the diagrams that you see in books are really an oversimplification, but it allows us to get the main idea across. So oftentimes we deal with oversimplification when we look at diagrams, but the main ideas in these diagrams are here. So let's go over the main idea, and then we'll look at a few more details as we look at what's happening at the chemical synapse. So if you just look at this basic diagram, what is the idea behind release of neurotransmitter? Well, first off, there needs to be an electrical signal. So that's depicted by this red line here. So there needs to be an action potential signal. So action potentials are traveling along the axon. They're moving their way all the way down to the axon terminal. And that action potential is going to be the trigger for starting the mechanism to release neurotransmitter. So as the action potential signal gets down to the axon terminal, it's going to open calcium voltage-gated channels. When you open calcium voltage-gated channels, you can have an influx of calcium ions into the axon terminal, and that becomes the trigger for releasing neurotransmitter. So the calcium voltage-gated channels are depicted down here in this diagram. So they're all over the axon terminal. And so here we see calcium coming in. And that calcium influx is going to trigger these vesicles to merge with the plasma membrane of the axon terminal. And then we're going to have an opening of that vesicle so that we can have release of the neurotransmitter. So we have exocytosis taking place. And then once that neurotransmitter is released, it can interact with a receptor on the postsynaptic cell so that we can have communication. Once you bind to the receptor, you're either going to get an EPSP, an excitatory postsynaptic potential, depending upon what ions flow into the cell, or you're going to get an IPSP, again, depending upon the movement of ions. So you're either going to get an excitatory signal or an inhibitory signal on the postsynaptic cell. So let's go through the details in the chemical synapse. So what's happening when you get this electrical signal that's opening the calcium voltage-gated channels? So what we have essentially is we've produced neurotransmitters. Those neurotransmitters have moved down into the axon terminal. We know that they're stored in vesicles. And they've actually started to go through a docking process, if you will. They will form complexes with the plasma membrane. So that's actually shown on this diagram here. You can see some interaction here, getting close to the membrane. So there's definitely some docking that takes place on this presynaptic membrane, but there still has to be a signal that's going to allow for the fusion and exocytosis to take place. So what we need is that action potential signal. So the action potential signal travels down the axon and it reaches the axon terminal. Once the action potential signal reaches the axon terminal, we open calcium voltage gated channels. So lots of calcium voltage gated channels depicted in this diagram. When you open the calcium voltage gated channels, you're going to have an influx of calcium. So we can see the calcium entering into the axon terminal. The calcium then is going to interact with sensor proteins. So there are sensor proteins found within the axon terminal that bind with calcium. Good example of a sensor protein would be calmodulin. There's lots of calcium-calmodulin interactions within the human body. We also use a sensor protein called synaptotagmin, which will interact with calcium. And again, these diagrams oftentimes are oversimplifications of what's taking place. Once the calcium interacts with its sensor proteins, whether it's calmodulin, whether it's synaptotagmins, that complex, so once you produce this complex between calcium and its sensor protein, it's able to activate an enzyme called protein kinase. So protein kinases, I'll say PK, right? Protein kinases exist in an inactive form within the axon terminal, but once they interact with the calcium, let's say calcium calmodulin, once they interact with that complex, that inactive protein kinase can become active protein kinase. And so it's labeled here, active protein kinase. Protein kinase is a phosphorylating enzyme. So it provides energy to other proteins within the axon terminal. So again, this isn't depicted in this diagram, but it's showing you that you get an activation of protein kinase and it becomes a phosphorylator. It's going to activate other proteins. And so what is it doing? Well, one of the main things that it does is it activates a protein that's part of the vesicles. So the vesicles have a protein called synapsin. So if I put an S here, it's synapsin. And they're found in the walls of the vesicles that house the neurotransmitters. So those synapsin proteins, if I was to draw a bunch of synapsin proteins here, they're going to get phosphorylated. So we're going to add some phosphate to them. And phosphate represents energy. So when you start to energize the vesicle, that's going to assist the vesicle in completing its docking and fusion process. It takes energy for exocytosis. So calcium influxes into the axon terminal, activates a sensor protein like calmodulin. That complex turns on protein kinase. Kinases go on to provide energy, and that energy then allows for the exocytosis process to take place. Because remember, exocytosis is an active transport mechanism, and so it requires energy for that process to take place. So we have energized vesicles. Those vesicles are opening up along the plasma membrane of the presynaptic cell, and now we're seeing release of the neurotransmitter. So the neurotransmitters are moving out into the synaptic cleft. And so once they're in the synaptic cleft, they can begin to diffuse over to the postsynaptic cell. And that postsynaptic cell can be another neuron, it can be cardiac muscle, smooth muscle, glandular tissue, etc. Once the neurotransmitter diffuses across the synaptic cleft, it's going to bind to a receptor. So I'll add in some receptors here. So the neurotransmitter is going to bind to a receptor. And depending upon what that interaction is, we'll either get an EPSP, an excitatory postsynaptic potential, or we'll get an IPSP. So let's say it's a neurotransmitter that binds to a receptor and allows sodium ions to enter in. Well, sodium can cause a depolarization of the postsynaptic cell, so that would be an excitatory postsynaptic potential. Let's say the neurotransmitter that gets released allows chlorine to come into the cell. Well, if chlorine enters the cell, that has a hyperpolarizing effect, and so that would be an IPSP, an inhibitory postsynaptic potential. So we can either turn on or turn off the postsynaptic cell depending upon what neurotransmitter gets released and what receptors it's interacting with. In order to turn this system off, then you have to have inactivation of the neurotransmitter. And so there are mechanisms for inactivating the neurotransmitter. So let's finish up with inactivation of the neurotransmitter. So 3 things to consider when you need to turn off a chemical synapse. One of the things that can happen is there can be reuptake of the neurotransmitter, either back into the presynaptic cell or into glial cells within the system. So here we have exocytosis of the neurotransmitter, and the neurotransmitter has gone on to bind to receptors. So these are receptors on the postsynaptic cell. But one of the things that you can see happening is those neurotransmitters can either go back into the presynaptic cell, or they can be taken up by glial cells and then recycled. So that's one way to turn off the neurotransmitter, just reuptake back into the presynaptic cell or the glial cells. What's interesting is there are medications that are reuptake inhibitors. So you may be familiar with something like MAOI, a monoamine oxidase. An MAOI is essentially a monoamine oxidase inhibitor, the imines inhibitor. Monoamine oxidase is really a degradation type enzyme. So we actually use enzymes to degrade the neurotransmitters. But if you're not degrading the neurotransmitters or you're blocking the reuptake of the neurotransmitters, then you can increase the amount of neurotransmitter that remains in the synaptic cleft. So we can block the reuptake through reuptake inhibiting medications, or we can use medications like an MAOI, a monoamine oxidase inhibitor, which will stop the enzymatic degradation of the neurotransmitter. And all of that is to increase the amount of neurotransmitter available in the synaptic cleft. And so that can allow the neurotransmitter to continue to bind the receptors and provide your postsynaptic response. And so that could be beneficial to treat certain neurological disorders, depressions, different types of psychological disorders. You may need to alter the levels of neurotransmitters within the synaptic cleft. So you might look at SSRIs, serotonin-specific reuptake inhibitors, or you might look at MAOIs, monoamine oxidase inhibitors, so that you can basically block the reuptake. SSRIs block the reuptake of serotonin. MAOIs prevent the degradation of serotonin, and that can all increase the amount of neurotransmitter. You certainly wouldn't want to mix those medications together, so you want to be careful about taking an SSRI along with an MAOI, because you could have an unsafe level of neurotransmitter within the synaptic cleft if you were taking both of them at the same time. So the second example of stopping the neurotransmitter is this idea of enzymatic degradation. So I was just talking about that a little bit. So here we have an enzyme that's being utilized to degrade the neurotransmitter when it's in the synaptic cleft, or possibly when it's in another cell as well. So you may have enzymes for degrading serotonin, like in MAO, a monoamine oxidase. You could have an enzyme for degrading acetylcholine. So you have an acetylcholinesterase, which is an enzyme used to degrade acetylcholine. There's COMT, catecholomethyltransferase, which is another degradation enzyme. And so all of these enzymes can be used to degrade the neurotransmitter and keep or basically stop the chemical synapse, okay? Lower the levels of neurotransmitter within the synaptic cleft. So this is an example of acetylcholinesterase. You can utilize that particular enzyme to degrade acetylcholine, turn it back into acetate and choline, and then you can have reuptake of those parts. And you can reuptake those parts and actually put the acetylcholine back together and utilize it again. But the idea here is that we use enzymes to decrease the amount of neurotransmitter in the synaptic cleft. The third way to stop the neurotransmitter is really just simply by diffusion. So the neurotransmitter can actually diffuse, let's say, into the interstitial spaces, or can diffuse into the circulatory system. And so as it diffuses away from the receptors, you're going to diminish the effects of that chemical on the postsynaptic cell. So what we've looked at is electrical synapses and chemical synapses, and we've also looked at ways to turn off the chemical, either through reuptake of the neurotransmitter, enzymatic degradation of the neurotransmitter, or simply by diffusion away from the synaptic cleft. So I hope that helps. Take care.